Hello everyone, welcome to my YouTube channel Pharma Researcher. So this is a video number 15 of Telangana Pharmacist Grade-II exam preparation. The exam is going to be held on the 30th of November 2024 and this is a series on our YouTube channel Pharma Researcher in which we are discussing a total of 10 units which are mentioned in the syllabus, and in these 10 units we have already covered the initial 7 units. Now only 3 units are remaining, and today we are starting to cover the unit number 8. So in this particular unit it is basically related to the pharmacognosy subject. So different multiple subsections are there. So I have divided this particular unit into 3 parts because the data is very large. So I have divided this particular unit into 3 parts. First part is this one, and the next part will be coming tomorrow, either 10 or 11 in the morning, and the third part will be coming tomorrow only in the evening. So by tomorrow we will be completing the unit number 8 also. So for the PDF, so PDF links are provided with you in the description box. You can download the PDFs as well, and if you haven't watched the previous videos, so you can also watch the previous video. All the links has been provided in the description box with all the parts, so you can watch those videos, you can revise the topics. So let's start with the syllabus discussion, like what different topics are there mentioned in the unit number 8. So basically this is the unit number 8, in which the first topic is regarding the adulteration and drug evaluation and significance of pharmacopoeial standards. Then the second part is the identification test, therapeutic effects, and pharmaceutical applications of alkaloid, terpenoids, glycoside, volatile oils, and tannins and resins. So these two topics will be covered in this particular video. In the next video, we will be covering these different topics like the occurrence, distribution, organoleptic evaluation, chemical constants, including the test of following categories of drugs. So these are the different categories in which there are several drugs. So I will include all the drugs from the diploma level syllabus because this particular syllabus is relevant to DPharm only. So then the pharmaceutical aids, in which different drugs are there. So this topic will be covered by tomorrow morning, and the miscellaneous and study sources of fibers that are used in sutures and surgical dressing. This topic will be covered in the third part. So by this way we will be completing this particular unit, and on Saturday we will be covering the unit number 9, and on Sunday we will finish our unit number 10, and with that we will be finishing the whole syllabus. So let's start with the first topic, that is the adulteration, drug evaluation, significance, and pharmacopoeia standards. So in this, the first question is, so there are very important definitions regarding drug adulteration. So different terms can be used for adulteration or substitution. So these will be covered in the form of MCQs. So the first question is, drug degradation by a substance resembling the genuine drug morphologically as well as organoleptically. So if the drug is being degraded by the addition of any substance which is resembling the genuine drug or which is similar looking, uh, with their morphological and organoleptic properties are somewhat similar to the original drug, so this is called as a adulteration. So here option C is correct. If anyone is talking about the substitution, so substitution term is not used for the resemblance. It is also used for the substitution of substance, but the whole substance is a different one. So that is the substitution thing. Then moving to the next question. Question is, dash is the admixture or substitution of genuine drugs with spurious, inferior, defective, useless, or harmful substances. So it is a admixture or substitution. So substitution of genuine drugs only, but it is with the spurious, inferior, or defective, useless, harmful substances. Okay, so it is basically almost similar, but it has no properties, it can cause harmful things, so it is also known as the adulteration. So here option A will be the correct answer. So in the later question, you will be having the definition of substitution. Then you will be clear like what is the difference between adulteration and substitution. Next question is dash is impairment of drug quality. So impairment of drug quality is the deterioration. Term used is deterioration. So here option D is correct. Next question is dash is defined as intentional type of adulteration. So intentional type of adulteration is sophistication. So here option C is correct. Next question is dash is addition of an entirely different substance instead of the original drug. So what is it is the addition of entirely different drug, different substance. Means we are replacing the original drug with entirely different substance. So that is known as the substitution. So in the adulteration, we are basically substituting the drug with similar looking substance that is resembling the drug, or similar looking substance which is having low quality. So this is the adulteration. If we are changing the entire different substance, if we are using the entire different substance for adulteration, that is the substitution. Then moving to next question. The question is dash is addition of any substandard drug. So addition of any substandard drug is inferiority. So here option A is correct. Next question is dash is accidental addition of one object into another due to ignorance or carelessness. So basically it is the adulteration only, but it is the accidental addition. Like we haven't added the substance on purpose, but it is accidentally added uh, due to the ignorance or carelessness. So this is known as admixture. So here option D is correct. Next question is dash occurs as a result of microbial attack. So spoilage is the term which is basically spoilage occurs as a result of microbial attack. When microbes attack on certain substances, then that substance or that material will spoil. Next question is if appropriate conditions of collection, transportation and identification are not maintained, accidental adulteration can occur, which is termed as. So basically there is a condition like if during the collection, transportation and identification, if we are not considering the appropriate conditions, we are not maintaining those conditions, then there can be accidental adulteration can occur during the process, during the transportation or during the identification time. So for this we are using a term undeliberate adulteration. So here option D is correct. So there are different terms with adulteration related things. So mostly all of the terms have been covered. So you can revisit these questions again. You will identify like for what term we are using, what type of conditions are there, what is the definition. So these are important. Then moving to next question. Question is evaluation of a drug involves determining its color, odor, taste, size, shape, touch, texture, etc. So these all the things are evaluating in the organoleptic properties. So here option B will be the correct answer. Then moving to next question. The question is dash is a qualitative evaluation type in which morphological and sensory characters of drugs are studied. So morphological and sensory characteristics are studied in organoleptic, same like color, odor, taste, shape, size. So these all things are organoleptic one. Next question is dash are of different forms and are used by the plants for storing food. So basically in plants the storage of food is basically done in the underground structures. There are some underground structures in the plants, and they use these structures, or these are of different forms. Underground structures are of different forms, and basically the plant's food is stored in these type of structures. These are the underground structures. Next question is Dash are the reproductive plant parts found in a variety of forms and also grow on the plant in different manners. So basically, these are the flowers. Flowers are the reproductive plant parts, and these are present in different variety of forms, and these are grow on different plants in different manners. So here the option C will be the correct answer. Next question is, seeds are characterized by a scar left by the funicle called. So basically in seeds there will be a scar which is left by the funicle when we remove that. So that scar is known as hilum. So basically if you have seen like different fruits and different vegetables like if you have seen mutter. So in mutter there is a this type of and there is a scar in the central part. So that scar is known as helium. Then next question is, seeds are characterized by a small hole in the ovule marking the entrance point of the pollen tube during fertilization called. So seeds which are characterized by a small hole, there is a small hole in the ovule which mark the entrance point, which is basically the entrance point of the pollen tubes during fertilization. So basically when in plants the fertilization occur, so pollen tubes or pollen grains will enter the ovule part, and in ovule there is a marking entrance point. So that is present in the seeds, and this point is known as the micropyle. So here option D will be the correct answer. Next question is, seeds are characterized by a vascular tissue ridge connecting the helium and chalaza called. So basically seeds are characterized by a vascular tissue ridge, and this vascular tissue ridge is connecting the hilum and chalaza. Basically what is chalaza? So it is simply the tissue where the nucleus and integuments of ovules join together. Okay, so nucleus and ovule main part are joined together. This is the chalaza. So these are joined at the raphe. So here option C is correct. Next question is, dash is the average number of stomata present in per square mm of leaf epidermis. So there is leaf epidermis, and in leaf epidermis at per mm square meter, uh, the number of stomata present in this is known as the stomatal number. If they are talking about the number, so it is a stomatal number. The next question is, dash is the percentage which the number of stomata forms to the total number of epidermal cells. So this is the epidermal cells. There are multiple epidermal cells, and in this epidermal cells the total number of stomatas formed in this to the total number, this is the percentage. In this percentage will be known as the stomatal index. So if talking about per mm square, in the area of per mm square the number of stomata present, that is a stomatal number. If it is talking about the like what percentage of stomata are present in the whole epidermis, so that is the stomatal index. Next question is, dash is a minute epidermal opening having a central pore and two kidney shaped guard cells containing chloroplast. So these are the stoma or stomatas. So basically there are two guard cells and multiple or a fixed number of subsidiary cells are arranged in this way, and in the plants these are the epidermal openings for basically the respiration and transportation work. Next question is, stomata are absent in. So in which part of the plant stomatas are absent? So these are present in fruits, flowers and stems, but these are absent in roots. So here option B is correct. Then next question is, in which of the following plant the stomata are present on both the surfaces? So the plant is senna. Senna is a type of plant which has the stomata in both the surfaces. In the inner, like if you can see the leaf, so in the upper portion and the lower portion, both the side, the stomatas are present. Next question is, dash stomata have two guard cells covered with two subsidiary cells whose long axes are arranged parallelly to the stomata. So basically these stomatas are of different types, and if like there are two guard cells, in this manner there are two guard cells, and they are saying there are two subsidiary cells, and these subsidiary cells are arranged parallelly. Parallelly means these are arranged in this manner, in the parallel manner. So these type of stomatas are known as parasitic stomata. So you can remember this why P is for parasitic, P is for parallelly. So these are the parasitic stomata. Now there can be another name for parasitic stomata that can be asked. So parasitic stomata is also known as ruviaceous stomata or parallel celled stomata. So parallel celled mostly will not be asked because it will reveal the answer. Mostly parasitic or ruviaceous, these two terms are mostly used. Then we can move to the next question. Next question is, dash stomata have two guard cells covered with two subsidiary cells arranged at right angle to the stoma. So now the similar condition is there. There are two guard cells. There are two subsidiary cells, but these are arranged at the right angle. At right angle means in this way. So these are the guard cells, and these outer portions are the subsidiary cells. And these subsidiary cells or these type of stomatas are known as diacytic stomata. These are known as diacytic stomata. So here option C will be the correct answer. And in this there are another names also. So diacytic stomata, it is also known as caryophyllaceous stomata, caryophyllaceous stomata, or it is also known as cross-celled stomata. And here the very important example is Vesaka. Vesaka is the example. Vesaka has having diacetic stomata. So for parasitic stomata, we miss the example. So you can remember the example like that parasitic stomata is present in cocoa, or it is present in the senna leaves. So mostly senna is asked previously in different question papers as well. So it becomes very important. Then moving to next question. The question is, dash stomata have two guard cells covered with three subsidiary cells. So basically these type of stomatas again having two guard cells, but there are now three subsidiary cells. So three subsidiary cells are basically in this way. So there is one another condition in this, like there are three subsidiary cells, but the third subsidiary cells is comparatively lesser in size than the other two. So these type of stomata are known as anisocytic stomata. So here option B will be the correct answer. And these anisocytic stomatas are also known as cruciferous stomata, or unequal celled stomata. So these are the different names. Now important thing is the example. So in anisocytic stomata, the examples are like Belladonna, Datura, there is Stramonium. So these are the different examples of anisocytic stomata. Next question is, dash stomata are surrounded by variable number of subsidiary cells. So basically we have two guard cells, and there can be variable multiple number of subsidiary cells. So these type of stomatas are known as anomocytic stomatas. So here option C will be the correct answer. And in anomocytic stomata, it is also very important, and these are also known as Ranunculus stomata, or irregular celled stomata. If you learn the basic things like the number of subsidiary cells and the guard cells arrangement, then you will be able to remember the names as well. In anomocytic stomata, example is very important. So example include digitalis. Many times this anomocytic stomata is asked for digitalis, and other examples include lovelia and buchu. So these are the several examples of plants containing anomocytic stomata. Then moving to the next question. The question is, which of the following plant has anomocytic stomata? This we have discussed. This is the digitalis. So here option D is correct. The next question is, dash is a plant having anisocytic stomata. This is the Tura. So here option B is correct. Then next question is, coca leaves and senna leaves have dash stomata. So coca and senna, these are having parasitic stomata. Then next question is, dash are tubular or elongated or glandular outgrowths of epidermal cells. So these are basically the trichomes. Basically on the surface there is something hair-like structures are present. So these hair-like structures which are tubular or elongated or glandular outgrowths. So these are the hair-like structures known as trichomes. So here option B will be the correct answer. Then moving to the another question. So the question is, dash are water absorption or secretion organs developed in some particular plants. So the water absorption and secretion organs which are developed in only in some particular plants, these are known as hydathoids. So here option C will be the correct answer. Then moving to the next question. The question is, dash method is an inexpensive analytical technique used for powdered crude drugs. So powdered crude drugs, there is a inexpensive analytical technique, and this technique is known as Lycopodium spore method. So here option D will be the correct answer. Then moving to the next question. The question is, alkaloidal bases are soluble in dash while alkaloidal salts are soluble in polar solvents. So basically the alkaloidal bases, these are soluble in the chloroform, but the alkaloidal salts, these are soluble in the polar solvents. So here option A will be the correct answer. Both things you need to remember, like bases, these are soluble in chloroforms and the salt forms are soluble in the polar solvents. Next question is, dash evaluation is a chemical process of determining the active constituent in a drug. So these are the chemical evaluation methods are those which involves the chemical processes to determine the active constituents in a drug. So here option B is correct. Next question is, dash is the measure of the capability of sample under test to produce biological effect in comparison to the standard preparation. So these are basically the bioassays in which we measure the capability of the sample under test to produce the biological effect in comparison to the standard preparation. So there is a standard preparation B, and there is a sample preparation in which we need to check whose ability or capability needs to be checked. So bioassays are carried out for these tests, and then the results are compared. Results are compared with the standard preparation, and these type of tests are known as bioassays. Next question is, in dash method, a solid nutrient medium is poured in Petri plates and inoculated with a microbial culture. So these are basically the cylinder plate method. So in this method, there is a Petri plate. In Petri plate, a solid nutrient medium is poured, and several microbial culture is inoculated in this. So I think this method we have also seen in the that social pharmacy part, also unit number 3. So there were different methods like streak method, single addition method. So those are the different methods. Now moving to the next question. The question is, anisocytic stomata are present in. So anisocytic stomata, these are present in the dura. So here option A will be the correct answer. Then next question is, Nux vomica seed contains. So basically Nux vomica seeds, they contain the unicellular lignified covering trichomes. So here option A will be the correct answer. Next question is, T-shaped trichomes are present in. So T-shaped trichomes, these are present in pyrethrum. So here option D will be the correct answer. Next question is, candelabra trichomes are present in. So candelabra trichomes are basically present in Varvascum thepsus. So here option A will be the correct answer. Next question is, stellate trichomes are present in. So stellate trichomes, these are present in Hemelis. So here option C will be the correct answer. So with this question we are done with this particular topic. Now we can move to our another topic, that is the identification test, therapeutic effects and pharmacological effects and pharmacological applications of alkaloid, terpenoids, glycosides, volatile oils, tannins and resins. So basically in this particular topic we are discussing the identification test. Various other like the therapeutic effects, the pharmaceutical applications of these compounds, these will be covered in the next part, in which we are covering the uses, biological source and other things as well. So here we are going to discuss the chemical test. I have provided you the theoretical part as well for different tests, so you can read that theoretical part. I will mention you like which of the tests are very very important. So in that way you can prepare. So let's start with the first question. So the question is, which one of the following is the basic chemical test to identify anthracene glycosides in alloys? So the basic chemical test to identify anthracene glycosides is modified von Traeger test. Basically, the alloys contain C glycosides. They contain C glycosides, and the C glycoside present in alloys is alloy imodine. Alloy imodine. So this test is basically used, and modified von Traeger test, this is used for C glycosides. That's why it is a test to identify anthracene glycosides in alloys. If I'll talk about the von Traeger test, so in von Traeger test it is used for O glycosides. O glycosides, in which the examples can be Sena. Then Keller-Kilani test, so it is used for deoxy sugars or the cardioglycoside, or it is a test for cardioglycosides, for example digitalis. Then there is legal test. So legal test is also for cardiac glycosides. Then moving to next question. The question is, when ERG's test is used to identify. So when ERG test, it is used to identify the ergot alkaloid. So here option C is correct. So basically in ergot alkaloid, the when ERG test is used, and sometimes the composition of when ERG reagent is asked. If asked about the when ERG's reagent composition, then the composition is para dimethyl amino benzaldehyde. This question is also asked in some of the exams, the composition part. So chemical tests are very very important. One question definitely will be there from chemical test. So you can remember this reagent composition. It is para dimethyl amino benzaldehyde, and it will give blue color with the ergot alkaloid. Next question is, Thalloquin test is used to identify. So Thalloquin test is used to identify cinchona, which contains quinine and quinidine, main important chemical constants. Quinine is Antimalarial, quinidine is antiarrhythmic. Now talking about the Thelequin test. So in this Thelequin test, we will take the powder drug. Powder drug is taken and it will react with bromine water. Bromine water. We will add bromine water and dilute ammonia solution. And dilute ammonia solution will be added. Now this powder drug will give emerald green color. It will give emerald green color with bromine water and dilute ammonia solution. So this is the Thelequin test. All are the important things, like for what drug Thelequin test is performed. It is for cinchona. What color will be produced in Thelequin test? It will be the emerald green color. What is the composition? Or sometimes the question can be like if cinchona is added with dilute ammonia or dilute ammonia solution or bromine water. Then what is the test? So in each and every aspect the question can be asked. So this is important. Next question is Keller-Killiani test is the identification test for. So Keller-Killiani test, it is the identification test for digitalis. So here option A is correct. Then next question is Mayer's reagent contains. So the Mayer's reagent is a test for alkaloids. So the composition of Mayer's reagent is potassium mercuric iodide. From M for Mayer's, M for mercuric. With this M you can remember the composition. Then there is potassium bismuth. It is the Dragendorf's reagent, also test for alkaloids. Iodine potassium iodide. So iodine potassium iodide is the Wegener's reagent. Wegener's reagent. And then there is picric acid. So it is the Hager's reagent. Then moving to next question. The question is which test is used for the identification of caffeine? So for the identification of caffeine, the test used is Murexide test. So Murexide test is basically a test for purine alkaloids. It is a test for purine alkaloids which contains caffeine as well as tea. So then coming to next question. So the question is which of the following active ingredients forms a froth when shaken with water? So the active ingredients which forms froth when shaken with water are saponin compounds or saponin glycosides. So here option C is correct. Next question is Dragendorf's reagent is used for detection of alkaloids. Chemically, Dragendorf's reagent is, so Dragendorf's composition is potassium bismuth iodide. So here option A will be the correct answer. Then moving to the next question. So the questions are almost over. So these are only few questions. Now I have added the identification analysis of phytoconstants in which different tests are provided to you. You can read these tests. I'll tell you the important ones and the rest you can read for your better understanding. So for alkaloids, there are different tests. Dragendorf's reagent, Mayer's reagent, Wegener's reagent, Hager's reagent, tannic acid, picolinanic acid, and Murexide test, and several specific tests for specific compounds. So in these tests, you need to remember the composition and the outcome, basically the color or the precipitate, these type of things. So for Dragendorf's reagent, the composition is potassium bismuth iodide and the color produces reddish brown precipitate. So these all data has been taken from the Kokate book. So you can, if you have the book, you can refer to that book as well. Then the second is Mayer's reagent. It will produce cream color precipitate and the composition is potassium mercuric iodide solution. Then the Wegener's reagent, it will produce a reddish brown precipitate, same as with Dragendorf's reagent, and the composition is iodine potassium iodide solution. Then there is Hager's reagent, so it will produce pale yellow precipitates and the composition is saturated solution of picric acid. So these four are very important. Mostly the question asked from Mayer's and Dragendorf's reagent most of the times. But in few question papers, the question has come from Wegener's as well as Hager's reagent. All the four are quite simple, so you can easily remember these. Then we can move to the next test, that is the tannic acid test. So it will produce buff color precipitate, not that important. Then the picrolinic acid test, it is also not that important, but you can always remember the colors because the similar type of things are there. So it will produce yellow color precipitate. Then the murexide test, it is for purine alkaloids like tea and caffeine. So this is important with respect to examples. Color you can also learn like caffeine is basically taken in petri dish to which hydrochloric acid and potassium chlorate are added and heated to dryness. Purple color is obtained by exposing the residues to vapors of dilute ammonia, and then after some time the purple color will be lost. So this is the murexide test. So color can be asked like purple color is produced in this test. So these things can be asked. Then there is Van Ark's reagent, important one. I have shown you the question as well. The composition you can remember dimethylamino benzaldehyde, para-dimethylamino benzaldehyde, and it will produce blue color. The next question is Vitali-Morin test, also important. Sometimes the question is asked like which test is for the tropane alkaloids. So this is the Vitali-Morin test. Examples you can remember like Belladonna, Datura, Hyoscyamus, Strontium, Devoisia. So these all are the tropane alkaloids, and the identification test is Vitali-Morin test. And here the violet coloration is produced, so color will be violet as an outcome. So this thing you can remember. Next is Theleguin test. So it is for cinchona. It will produce emerald green color with bromine water and dilute ammonia solution. These things you can remember. Then there are several general tests for glycosides, test A and test B. These you can remember. Mostly the specific test will be mostly asked in the examinations. So first is for specific glycoside, that is the anthraquinone glycosides. So here the bond trigger test for O glycosides, modified bond trigger test for C glycoside. This we have discussed in the first question. Then there is some another test, non-naming test. What very rare chance like these type of test will be asked. But you can simply remember like which reagents are reacting and which color is produced, so that you can see. Then next is a test for hydroxyanthraquinones. So there is no specific name for the test for hydroxyanthraquinone. What you can remember, like you can read once. So if the question comes from this particular test, which is very rare chance, but you will be able to memorize few of the things. Then next is for cardiac glycosides, that is cardinalides like digitalis, strophanthus, thevetia. So there is Lieberman-Woodchar test, that is test for bufordinolides, which is the squill. So this question can be asked. Okay, so the identification test for squill. So again, I'm saying this is rarely asked, but this can be asked. Mostly in this, the test asked were Keller-Kiliani test for digitalis or deoxy sugars. So these things are asked because if you can see digitalis, so digitalis is a very important drug because it is used in cardiac conditions, and cardiac conditions are a very serious condition, and very high percentage of peoples are catching up with these cardiac conditions. So that's why in the daily need and in the society there is a need of these type of drugs. So that's why these drugs becomes very important. Then other tests are like Raymond's test, legal test, Walger test. Uh, then in these also you can remember the names at least for which particular drug it is used, like Raymond test, legal test, Walger test. These are for digitalis glycosides, not for exactly for digitalis, but these are for anthraquinone glycosides. Okay, so these not for anthraquinone, these are for cardioglycosides. For anthraquinone glycosides, there are von Traeger and modified von Traeger tests. So these different tests are for digitalis glycosides. Next, we can move to the coumarin glycosides, in which there are Soralia, Visnaga, Me, Cantharides, Cochineal, Milavres. So this test you can read from here. Then again, there is cyanogenetic glycosides. There are several. So basically, here only the procedures are mentioned. There is no specific naming test for these compounds. So the tests which are with the name, those are very important. For example, this for saponin glycosides like Brahmi, Diascoria, Ginseng, Gokru, Jal Brahmi, Quellia, Sinega, Shatabri, Yasti, Safed Musli. For these there are two tests. One is froth formation test. So this question I have given you in the MCQs also. Like for froth formation test in which the froth is formed. So those are for saponin glycosides. Also there is hemolysis test. So hemolysis basically in this, the rupturing of blood is seen with these type of glycosides. So these are basically two tests for saponin glycosides. Then there are a few tests for terpenoids and volatile oils. These are quite simple. So, previously the question has been seen from the Sudan 3 test, like regarding the color as well as the test name also. Like Sudan 3 test is for which type of secondary metabolite, so it is for terpenoids or volatile oils. Then there is tincture of alkana test, so this is also for terpenoids and volatile oils, and in both these tests a red color is obtained. Then coming to next, so these are the steroids and triterpenoids. So for steroid and triterpenoids, there is Lieberman-Burchard test and Selkovsky test. So here also you can remember the name. Specifically, sometime asked for the triterpenoids. So these two tests you can remember. You can read these theoretical parts and memorize these things. So that is the best way. So yeah, these two topics have been covered. I have provided you the text, I have provided you the MCQs form. Both the PDFs will be provided to you. Next, we will be going to discuss the next part, that is the organoleptic properties and chemical constant biological source of different classes of drugs. That will be very, very important. So that's all for this particular video. Let's meet tomorrow. So till then, thank you very much.